Thank you. Can everyone hear me at the back? Okay, I'm a postdoc in David Kingsley's lab at Stanford University. Today I'm going to tell you a story about some of the work we've been doing on parallel evolution between marine and freshwater sticklebacks. I want to start by thanking the coordinators for inviting me for the opportunity to present this work. And I also wanted to acknowledge that the work you'll see today is actually a huge team effort, and it's work done by multiple different labs in multiple different universities, including Dmitry Petrov's lab at Stanford University, the Kingsley lab where I'm from, Sherston Lindblad-Toe's lab in the Broad Institute, and Rick Meyer's group at Hudson Alpha Institute. So many, many players, and one of the players who's played a big role in the genomics that you'll hear about today is Frank Chan, and he's here in the audience. So definitely an equal part effort between Frank and myself for some of the work that you'll see today. Okay, so you've already heard a lot about sticklebacks, and I'm somewhat intimidated by having to follow such awesome talks, but here's my attempt. There is a massive amount of diversity in sticklebacks, and you've heard about limnetic and benthic forms and also lake and stream forms. Much of this diversity has arisen since the ancestral marine stickleback invaded multiple different freshwater habitats, and so we see a whole bunch of different freshwater forms, and that's a typical adaptive radiation like a starburst phylogeny. Much of this has occurred in the last 10,000 years since the retreat of the ice sheet across the northern hemisphere. So the ancestral marine fish invaded new empty habitats, and they adapted to their new habitats. I'm going to tell you about the marine freshwater aspect of this divergence, and so I'm going to start by giving you an ecological angle on this story, and then I'm going to switch halfway through to talk about genomics. And the ecology is work that I did during my PhD in the University of Edinburgh, so it's quite old. Bear with me, it's been a while since I've talked about it, but here we go. So the marine and freshwater forms come into contact in the lower reaches of estuaries throughout the world. So marine fish actually are, what we call marine fish, are truly actually anadromous fish like salmon. They actually breed in these lower reaches of the rivers in freshwater. They migrate out to sea and they come back to breed again. So when you look at this contact zone between marine and freshwater forms, you can find that they're coexisting in sympatry. And this is co-occurring along an environmental gradient in salinity and a whole bunch of other environmental parameters. Just to give you a feel for what the sympatric sites look like, I'm showing you here some morphometric data, but the genetic data looks very similar. As you move from the marine sites at the coast to the freshwater sites upstream, you can see in the lower reaches of the river where there is this salinity gradient, two divergent morphometric morphologies that exist in sympatry, and the genetic data looks very similar. So we have good evidence that there's distinct clusters that coexist, and actually if these are adults sampled from the hybrid zones. If you look at the juveniles that they produce, you can see that there's quite a few hybrids produced, but the hybrid zone is mostly a bimodal type of zone. So given that there is this opportunity for gene flow, one of the questions that we'd like to know is, what maintains the distinctness between the forms. So I'm going to briefly run through what we think might play a role. We do have some evidence initially for in the sites of sympatry there is temporal displacement in breeding. So the blue line here represents the peak of the breeding season for freshwater fish. The red line represents the breeding season for marine fish. So they are slightly displaced, but you can see that there's at least 3 months of overlap where these forms in sympatry have the opportunity to hybridize. So although there is displacement, it's not complete. In replicate pond experiments, I was able to show that there's no evidence for assortative mate choice. The assortative mating coefficients in these were not significantly different from 0, so we have no evidence that mate choice plays a role in this particular system. And because of that, we see actually that pre-mating isolation is not strong. Hybridization is very common, and when you look at the individuals in the middle of the hybrid zone, we see a huge number of hybrids and backcrosses and recombinants. So that means that speciation or whatever is definitely not complete, but there's still something that must be contributing to the ability for these forms to remain genetically and morphologically distinct. To go into the post-mating barriers, we see no evidence of genetic incompatibility, so if you raise the individuals in the lab, F1s are totally fit and just as fit as the pure forms. However, we do see evidence for disruptive selection in the wild, so this is 2 examples of that. On the left here, I'm showing you body size on the y-axis as a proxy for fitness. And on the x-axis here, I'm just showing you lateral plate morphology, which is a trait that differs between the marine and freshwater forms. And you can see here that individuals of intermediate morphology are definitely smaller in size compared to the pure freshwater forms and the marine forms. And this difference, although the axes I noticed fell off, this difference is about a difference of 4 to 5 mil, and for sticklebacks, that's just a little bit less than 10% in body size. Similarly, on this side of the plot, I'm showing you the probability of overwinter survival. So I actually tracked individuals over time in the hybrid zone, resampling, and I was able to show that the probability of overwinter survival was significantly related to the genetic ancestry. Individuals of intermediate genetic ancestry were much less likely to survive winter than the freshwater or marine forms. So we've got good evidence that there's strong disruptive selection going on in this system. And we think that actually the divergence between the forms is actually mostly ecologically mediated by these processes, and that much of reproductive isolation in this system is actually just gene by environment interactions operating in these fashions. So just to summarize the quick summary I've given you, we find no particular strong pre-mating isolation. They're not spatially isolated. There's some displacement in temporal in breeding season, but it's not complete. There's no mate choice, assortative mate choice. There's no genetic incompatibilities, but there is hybrid inferiority. Okay, so if we really want to understand the reproductive isolation that's maintaining differences here, we would like to know how genes and the environment interact to influence this hybrid inferiority, influence the fitness. And there's been a lot of discussion, apparently, not that I was involved in it, but whether the genetics of adaptation actually is the same as the genetics of reproductive isolation. And I think in this particular system, if you believe that reproductive isolation is driven by hybrid inferiority, then the genetics of adaptation would be equivalent, because we're looking at how genes and the environment interact. So step one, I'm going to switch now to the genomics side of the project. It's a slightly different story. However, I think that it does inform us about the genetics of reproductive isolation. So I'm going to describe to you work that we've been doing in the Kingsley lab. And we're looking at, mostly interested in mapping the genetics of adaptive evolution. And I think that it does tell us quite a bit about the genetic basis of reproductive isolation. So the project I'll tell you about is understanding the genomics of parallel evolution between marine and freshwater forms. I showed you earlier a starburst radiation of sticklebacks from the ancestral marine form. A lot of the changes that have happened have also happened in parallel. So there are some radiation patterns, but there's also parallel changes. And in this particular grisly plot of dead sticklebacks, you can see some of the differences that have evolved in parallel. As you look across the world, marine fish are consistently larger in body size, they have more armour, they are more robustly shaped compared to their freshwater counterparts. So we see this repeated pattern of divergence throughout the world. There's been a lot of mapping done by various people in the Kingsley lab before me, and it's a huge amount of work and represents years and years and years of people's lives. However, we have had some success, and we have been able to identify genes underlying some of these adaptive traits. For example, KIT ligand underlying pigmentation differences, the EDA gene underlying differences in plate morphology, and PITX1 underlying differences in the presence and absence of the pelvic girdle and spines. We also have mapped QTLs, more than 100 of them, and we're at various stages of fine mapping these. And these are QTLs which have underlie mostly many different traits, actually, a lot of morphological traits, but also several other physiological traits as well. And they have varying effect sizes. But I wanted to emphasize at this point that taking this forward genetic approach and picking a divergent trait is not necessarily the same or equivalent to understanding, or it's not the best approach to use to understand the genomic basis of reproductive isolation. Because although EDA does differ between my forms, and there is differences in lateral plate morphology, it would not be sound to say that this is the single only reproductive isolating locus in the system. So using this kind of trait mapping approach is useful, but is not the same as actually understanding the divergence between forms in natural systems. However, there are things that we can learn about this. So all three of these traits have evolved in parallel in multiple different populations. And we do have evidence for selection in each of these situations. They also have characteristic genomic signatures, and with the advent of next-gen technology, we're actually able to ask, can we use these genomic signatures to find other genes that, that exist in the genome that underlie adaptive evolution? So we know about two possible modes of parallel phenotypic evolution in sticklebacks. One is via reuse of standing genetic variation. So a particular freshwater population might sweep to fixation an ancestral allele. It's the same allele that's swept to fixation in other freshwater populations, for example. And that allele is divergent and different from the ancestral marine form. This pattern of reuse of standing genetic variation, the same haplotype being swept to fixation across populations, is what we see for the EDA and KIT ligand pigmentation example. It contrasts to what we see for the genetic basis of repeated pelvic loss in stickleback. So in Frank's study, he actually was able to show that repeated pelvic loss involves a regulatory enhancer just near the PEDX1 gene. It's the same genetic basis insofar as it's the same genetic pathway, but actually each population that's lost its spine has its own independent unique mutation and has actually deleted the regulatory element in its own unique way. So there's no sharing of ancestral variation in this context. Okay, so I'll be right up front and tell you that it's very, very expensive genomically to look for this kind of signature. Today I'm going to tell you about a genomic project we're doing just concentrating on this parallel evolution via reuse of standing genetic variation. So I'm not even going to pretend that I'm catching this side of the pile. And to put this into context of Kirsten's talk yesterday, she described parallel evolution in 4 different categories, and this pattern of reuse of standing genetic variation that we see here would fit into her evolution in concert category, where we have effectively on a species level balancing selection. The repeated fixation of an adaptive allele in these populations is actually occurring because the allele is at low frequency in the marine population. It's been repeatedly swept to high frequency in these populations. Okay, so I'm going to tell you about genomic resequencing of 21 genomes of sticklebacks. We wanted to pick 10 marine and 10 freshwater sticklebacks from around the world and actually sequence to see if we could identify the genetic basis of parallel evolution. The marine freshwater hybrid zone I told you about earlier at the start actually is just one example of many contact zones that occur around the world. So we took advantage of this trait, this particular feature of stickleback biology, where we've got these pairs in contact with each other, and we chose, we wanted to choose populations that would represent typical marine and freshwater pairs. To do that, we actually started by using geometric morphometrics. The morphology or the morphometrics is a useful trait to pick typical freshwater and marine populations. We know it's got a genetic basis, we know it's polygenic, and so we can be confident that if we see a typical freshwater morphology that we're actually going to be catching something that's spread across the genome. So we started by performing morphometrics on something like 50 or 60 populations around the world. You can see the major axes of variation amongst all these populations. Each of these dots represents the centroid of a population, so there's actually many more individuals on here. It's just hard to see when they're all on top of each other. So I'm showing you the centroid of a population. The major axes of variation here is an axis which separates out the red marine fish from the blue freshwater fish. You can see there's a huge amount of morphometric variation in freshwater populations, but we wanted to concentrate on typical marine freshwater divergence. So rather than sequencing the genomes of some of these unusual freshwater fish, we actually concentrated on individuals from populations that were close to the ecotype centroid. And that's what I've shown in solid colors here. So the filled-in dots are the ones we actually went forward and sequenced using Illumina technology. I'm going to skip through quite a bit of the details, but some of the results I'll show you are based on analysis of sliding windows across the genome. And the sliding window size I'll be using is a 2.5 kb window. The after collecting 21 genomes worth of data, the approach we chose to take was actually a distance matrix approach. So for each window in the genome, we would build a pairwise genetic distance matrix comparing each fish to each other fish. So each cell in this particular matrix represents the pairwise difference between two fish, pairwise nucleotide divergence. And this is an example of the matrix that would come out of a window within the EDA region, which is, in a way, a positive control. You can see here that dark squares represent a lot of genetic divergence, and pale squares represent not very much divergence. So you can see there's a huge amount of structure that exists within this particular window. And this is really useful. I mean, anyone who's familiar with matrices knows there's many, many things you can do with it. So one of the things that we did with it was to do some multi-dimensional scaling and look at clustering or structure that exists within the matrix. We can actually compare what we see at a region like EDA to a region which is neutral or somewhere else in the genome. I think this is somewhere on chromosome 3. We see very little structure within the matrix, and when we do the scaling on this particular thing, we don't see much clustering. So we came up with a way of measuring the kind of clustering that we see within a matrix, and we call this the cluster separation score. It takes into account the variance that we see within a cluster and the variance that we see between clusters. So we can go in with a particular marine freshwater hypothesis and ask how much variance is there within the freshwater class, how much variance is there within the marine class, and how much variance is there between. And we come up with this cluster separation score that reflects the magnitude of clustering. And this is a really useful measure of parallel evolution, so it's actually reflecting this pattern of global reuse of standing genetic variation. It's a parallel divergence measure. Okay, so just to give you an idea of what we kind of see as we look across the genome, this is one of the chromosomes, chromosome 4. We can actually see really high resolution signals of marine freshwater divergence. 3 very clear peaks here, for example. And it's nice to see that, you know, some genes that we've already mapped independently and done fine mapping on actually come up as we would expect. So this particular peak corresponds to the EDA region. We see that there's massive cluster separation score directly above the EDA gene. It's actually quite an interesting topology around here, but the signal is centered upon EDA, and that's nice to see. But we also came up with these other regions that we had no idea about from our mapping. We had no idea that these exist, and we see very, very high resolution patterns of global reuse of standing genetic variation. So I'm showing you the raw data in the bottom here. The marine fish are in the bottom 10 rows, the freshwater fish are in the top 11 rows. And I've color-coded the alleles as red being the ancestral marine allele and blue being the derived allele. And you can see, just by eyeballing the raw data, that there is an excess of derived blue alleles in these particular regions. So it's a strong signature of a sweep that's occurred. And it's also obvious just from this region here that the size resolution is quite extraordinary. We see some very small windows. We also see some broader ones. So very interesting patterns of repeated selection here. On a genome-wide scale, this is the kind of pattern that we see. So I've just shown you these 2 examples on chromosome 4, but you can see across the genome there's many regions that show these kind of patterns. So the first conclusion from this is that parallel evolution of marine and freshwater forms does not involve a single supergene cluster. It actually involves multiple loci around the genome. And we're pretty excited about this because it actually provides us with huge numbers of things that we can chase up, and some of them are just so extraordinarily tight, and we could actually do transgenics and all sorts of really cool assays. So this is a very fun data set to play with at the moment. This is a continuous measure. So I was talking with Andrew yesterday about how important it is to recognize the continuous measure of divergence, right? However, if you just wanted to take a slice and ask how many genes are selected or not, here is a slice I took with a false discovery rate of 0.02. We see 42 distinct regions in the genome that are under selection that cover about 70 genes and about less than 500 kb in total, which is about 0.1% of the genome. So a very small representative of the genome is responsible for the divergence that we see between forms. All right, so I mentioned already that they vary considerably in size, but we were really surprised to see some of these are actually quite huge, like 400 kb in size. And with data I'm not going to talk about today, we have good evidence to suggest that some of this is associated with structural rearrangement. So inversions are actually trapping together marine freshwater alleles that are segregating across the world. That's exciting too. All right, and to step back again to start thinking about reproductive isolation, during my PhD, before the genome was sequenced, before we had any of this kind of data, I actually hand identified a number of polymorphic markers and diagnostic markers and was able to study the change in allele frequency across the hybrid zones. This particular marker was one of the best ones that I found. It was located in an ATPase gene, which is responsible for salt regulation in multiple different fish species. We could see that along this single environmental gradient, we could see that the frequency of the ATPase allele changes in a way that corresponds directly with the salinity gradient. So that was nice. It turns out that that particular SNP, or whatever I found back then, falls right here on this peak here. So it was really nice to see that during my PhD I was able to hit one of these regions. Okay, so how would we actually evolve this kind of pattern of parallel reuse of standing genetic variation across the entire world? The hypothesis that most of us work with in stickleback is what is known as the transporter room hypothesis. The marine fish exist and colonize freshwater habitats, and there's constant gene flow between the forms. We think that this gene flow is responsible for the leakage of adaptive alleles out of a freshwater habitat, and they're present at low frequency in the marine fish, which are then able to spread them around into the next door neighboring habitats. So actually we have some estimates about the frequency of various adaptive alleles, ranging from effectively close to 0% as far as we can measure, up to relatively high frequency alleles at 6%. So for parallel evolutionist sticklebacks, really this hybridization between the forms is quite key to the spread of adaptive alleles. Which brings me to the final part of the talk, which is thinking about when a single colonization event occurs, how much of the adaptation that occurs in the freshwater environment involves reuse of this standing genetic variation, and how much actually involves genetic variations independent to that particular population. So we can start to look at that question with this data set. I told you already how we've got this cluster separation score, which represents the globally reused standing genetic variation. So it's measuring parallel evolution across the world. We can actually just dive into one particular cell in the matrix, which represents divergence between a particular marine and freshwater pair on a local scale. And that represents the locally relevant adaptive divergence between those forms. And we can look at the relationships across the genome. So every window in the genome will have both a locally relevant measure as well as a globally relevant measure. So I'm going to show you what that looks like on this particular plot. So on the x-axis here, I'm showing local divergence. On the y-axis here, I'm showing global standing variation. And we'd like to know how much of this local divergence actually comes from the globally shared variation. This is what it looks like. The dots, each of these dots represents one window in the genome, and I've color-coded in red here just those windows that pass this false discovery rate of 0.02 for the globally shared variation. So you can see, if you see this part of the x-axis here, these are regions of the genome that are great for candidate reproductive isolation. Between a given pair, they're locally divergent and probably underlie the genes by environment interactions that I spoke about earlier. Some of that, actually a fair proportion of that, actually does come from globally shared variation. So we think that reproductive isolation between any given pair does rely quite heavily, or at least some degree, on globally relevant variation. And these are the candidate loci that we can concentrate on for understanding the genetics of reproductive isolation. So I'm going to zoom back out and put that into context now. The story I told you about with the genomics was really this pattern of reuse of standing genetic variation. I told you that we were not really looking at this pile of independent mutations, although just now with the local divergence we can get some idea of how much is going on in this side. To really address this pile of things, we need to have multiple individuals from each population, and we need to have very high resolution because we know that sweeps can be extremely small, and it's dependent on the age of the sweep and the recombination rate in the genome. So ongoing projects involve resequencing of multiple marine and freshwater individuals from multiple replicate hybrid zones. And I'm running way out of time, so I'm going to skip over a lot of this just to say that I've told you about the ecologically mediated selection against hybrids, how we can actually infer that some of these, the genetic basis of some of this is actually parallel. And finish by saying that actually we're expanding this hugely. So we're now doing resequencing on multiple different types of stickleback species pairs, and we're excited to be able to understand whether the genetics of reproductive isolation between marine and freshwater fish is the same as limnatic and benthic fish or lake or stream fish. And finally, I actually wanted to make sure that it goes back into the wild. So we've actually designed genome-wide arrays so that we can start looking at fitness landscapes across the genome for each of these regions. So I'll just stop there now and say thank you to all of these people. And that's it. Thanks.
Yeah, thank you, Felicity. I think we have time for one or two questions. Answer the first one. Right in front of you.
I enjoy your talk. I've been following your excellent work in the last 8 years. And the only thing that I would like to ask is, in this each one gene, which the evolution has been not in coding genes, but in the enhancer in the part, is it one of the best probably examples we have about how natural selection acts in the regulatory part of the genome.
Yes.
And there has been a lot of speculation about how natural selection is acting on developmental genes, you know. People don't believe that evolution in developmental genes is fooled by natural selection. They believe that it's just internal constraints that guide this in the parallel evolution, I should say. What do you feel about this? Is this fooled by natural selection? And what about the relationship between this speech one enhancer variability that you find with predatory effects?
All right, so I'll divide that into two kind of aspects, and Frank, being the PIDX1 person, can probably answer one of those more than I can. So to address the coding versus regulatory debate, we actually can look at these regions that we see for parallel evolution and split them up into things that fall within genes or in intergenic regions, and we have if you were to make that pie chart, we think that about 50% of these regions actually appear to be mostly intergenic, so not coding, and a very small percent actually seem to be coding-related changes that we can detect. So we have fair confidence that most of this parallel evolution is regulatory related. And then I'll pass the PIDX1 story over to you. So the question was actually asking, for the PIDX1 region, what was about the predation?
Whether it is fooled by natural selection, do you think, and what relationship is with predatory behavior, you know? Because they say that benthic and limnatic have evolved different pelvis reduction because of different predators they found. Okay.
In the interest of time, I would just very quickly say that the molecular variation, we're quite certain that it was under selection. There's a speed that we can detect very consistently. But then ecological hypotheses that are quite a bit of them. Right now, having the mutation allows people to completely go in and test it. And in terms of existing work, it's still subject to whether they have not been able to pin down exactly how ecologically these two as
in terms of the locally divergent elements,
yes.
have you any feel for regionality of this?
Right, so it may not be global, but it might be from the river next door, is it?
Regions of the world rather than...
Yeah, so actually we can start looking at, I've told you a story about one of the marine freshwater pairs as my local measure, but actually I can look at the one that's nearby and ask how much overlap there is there, and it becomes a multi-dimensional space. But certainly when you think about that scatter plot, a lot of the variation is shared. Whether it's all the way globally shared or if it's just next door, you know, is very rarely going to be a unique thing specific to that river system. And I think that just reflects stickleback biology. There is a lot of dispersal and shuffling of genes around. So.
Okay, thank you again.